Welcome, everyone, to the Weinberg Group webinar entitled Important Lessons in Developing Biosimilars. All participant lines will be in a listen-only mode throughout the call. However, if you would like to submit a question or comment to the presenters, click the button marked Ask a Question on your event window and submit your question. All questions will be answered via email within a week of the presentation. To start, I would like to turn the conference over to Michael Switt. Mr. Switt, go ahead.
Thank you so much. And good morning and good afternoon to all of you who have taken the time out of your day to join our webinar today entitled Important Lessons in Developing Biosimilars, sponsored by the Weinberg Group. We are a scientific and regulatory consulting firm that since 1983 has been helping bring highly regulated products to the marketplace both here in the United States and in Europe. And in that time we have significant experience in addressing issues relating to biologics and generic drugs, and therefore we're looking forward to sharing with you today some of our insights on developing biosimilars, particularly after the passage last year of the Biologics Price Competition and Innovation Act. I think you're really going to enjoy your presenters today. The two presenters in order of appearance will be Dr. Robert Roth. Bob is our medical director, and you have a short bio for him on the next slide. And joining him will be Dr. Nicholas Fleischer, who heads our clinical pharmacology and biopharmaceutics group. Nick had an outstanding career at FDA before joining the Weinberg Group in 1997, and in fact his last posting was at FDA was as head of the Division of Bioequivalence within the Office of Generic Drugs. One last thing before I turn the podium over to Dr. Roth is many of you will probably be wondering when will we get the slides. At the same time that we send out the questions and answers, we will also send the slide deck for you. So you will be getting the slides within a week of this presentation. So without further ado, I'd like to invite my colleague Bob Roth to kick off today's webinar.
Thank you very much, Michael. What I'd like to cover today, we'd like to start with a brief overview about what biosimilars are all about. I'm going to then spend most of my time talking about some scientific and product development issues, and then turn the presentation over to Nick, who's going to be focused mainly on discussion of regulatory issues, and then we'll finish up with questions. Now, for background, the follow-on biologics, the biosimilars field is a very, very significant part of modern-day therapeutics. Many recombinant drugs are coming off patent. The big-ticket items, drugs like erythropoietin, GCSF, granulocyte colony stimulating factor, the various insulins, monoclonal antibodies, interferons, these are all very well accepted therapeutic entities. They're proteins. Not all of them are biologics in a regulatory sense. Some were approved as new drugs. However, they're all biological in terms of their mechanism of activity. They're all proteins. The ones that we're going to focus on are all based on cloning technology. That means that these are proteins that are created not synthetically in a laboratory or by purification, but created in microbial manufacturing plants, if you will. And this field is really important. I mean, there's been many, many new therapeutic approaches that have been possible with the development of these types of agents, approaches that were not available 20 years ago, and clinical outcomes are improved as well. Some of these biological proteins that would be candidates for biosimilars are blockbusters in terms of revenue. Recombinant erythropoietin sells billions a year. And the concept that initially started out that of a biogeneric group of drugs was seen as a possible way to streamline the development process and reduce cost and ultimately reduce price to consumers. And the approvals of some of the initial biosimilars that we'll discuss in the next few slides have actually made this a reality. However, I should mention that the cost of development of these drugs is very large, and I'm going to spend a little bit of time talking about a few of the details related to the development programs and what their costs could be, and therefore the cost savings to consumers is not particularly great, certainly not within the concept of an old-fashioned generic drug. Should also point out this time, however, that these types of products, the therapeutic proteins, are extremely expensive to make and extremely expensive in terms of cost to the consumer. And so even a small percentage change in the cost to the consumer for a biosimilar product can translate into a significant reduction in costs in terms of actual dollars. Now, the approvals to date are limited. The field of biosimilar approvals began in Europe, and in the EU there have been 14 biosimilar authorizations to my count. This is a bit misleading in that there are several versions of the same product and under different trade names to make up this total of 14 biosimilar approvals. What they amount to are two versions of somatropin or growth hormone. They're not identical. 5 versions of erythropoietin and 7 of filgrastim GCSF. In the U.S., the 2 somatropin growth hormone products, omnitropin, valtropin, were subsequently approved years after the approval in Europe. Should also point out at this point that many of the products that are candidates for biosimilars or future candidates were approved as BLAs through Biologics. The approvals in the U.S. of the omnitropin, valtropin, the growth hormones, these products, the original innovator growth hormones, were actually approved as drugs, and so the biosimilar approvals were drug approvals under 505(b)(2) applications. Now, we got interested in the field about 10 years ago. There was a lot of interest raised in the media about biogenerics, or a cheaper version, a generic version of biological proteins. Many of the innovator companies and others prepared citizen petitions raising many of the scientific arguments and problems with the field, as well as the fact that at that time there was no ready regulatory pathway. Many of the issues that were brought up, were raised for biosimilars are the same today as they were a decade ago. Issues having to do with the sameness of the actual compounds themselves, the therapeutics themselves. This is a problem because they're large, they're very complex, many of them are glycosylated, the structure, the three-dimensional structure is critical, and these are all as much an issue today as they were before. There was always the question, well, how similar does the follow-on product have to be from the original? How close is close? And can 2 independently manufactured proteins actually be identical? I think the answer has turned out that biosimilar proteins can be close in terms of their characterizations, but not identical. And the bigger question really is, to what extent does it matter how close they are? And this is a particularly vexing issue for large proteins because much of a protein molecule is essentially irrelevant from a functional perspective, and so some small changes in some areas may be quite unimportant. Small changes in other areas can be critical. The other issue that's become clear is that assessing sameness between an innovator and a follow-on, a biosimilar version of a molecule is technology dependent. And as the science evolves and we get more and more sensitive and new ways of looking at structure, and especially three-dimensional structure, the assessment of sameness can change. I would say that in terms of prediction, if you look at the structure and composition of a biosimilar protein and do some laboratory tests of various sorts, maybe some cell culture animal studies, it's fairly straightforward to make some predictions about pharmacology and activity that may in fact hold up. It's much more of a problem when you're talking about safety and risks. Laboratory testing does not appear to be nearly as advanced in terms of predicting risk. I'd like to make a couple of comments about the active ingredients, the recombinant proteins themselves. In terms of a classical generic small molecule, these are relatively straightforward to make in terms of manufacturing, in terms of their chemistry and describing their composition, and therefore many manufacturing facilities are capable of making the same active ingredient. This is probably certainly not at this time the case for biosimilar proteins, not likely to be the case at least for a while. Technologically, it's much more difficult to make these products, to characterize these products, and there are fewer sources. It turns out to be quite difficult to match the three-dimensional structure of these biosimilar proteins, how they refold once they're manufactured, post-translational modifications like putting on carbohydrate. These are all sort of difficult to match from one center to another, and what becomes a major issue in terms of safety, it's quite difficult to match biological, the contaminants. Just as a reminder, contaminants in the world of a classic small molecule generic may be something like a solvent. Contaminants for a biosimilar that's made by recombinant technology would be things like if the protein is made in E. coli proteins, or E. coli carbohydrates, or E. coli DNA or RNA. These are different kinds of contaminants that were ever dealt with before. We think that it's going to be quite difficult to get acceptability much more difficult to get acceptability of active ingredients that come from abroad, and in particular, China, I think, is going to be in particularly great scrutiny. Many of you are probably aware of the heparin scandal from a few years ago, heparin not being a biosimilar, but some very small contaminating components from overly sulfated chondroitin components in heparin turned out to be of major clinical impact with very severe allergic reactions and a lot of FDA scrutiny. Now, the data packages that are required for approval of drugs, if you compare a generic and a biosimilar, they're really night and day. Generic drug approvals, as you probably know, require characterization so that you can say that the active ingredient is the same as the innovator, and pharmacokinetic equivalence, PK studies showing that the blood levels are equivalent. In biosimilars, the picture has been quite different. The data packages that have been developed for those biosimilar proteins that have been approved by regulators have extensive physical chemical characterization, a variable amount of preclinical data, including a fair bit of animal toxicology, very extensive clinical databases. I'll talk about that next. And at the end of the day, when you look at the data that were amassed in the applications that were made, the applications look like new drug applications, not like ANDAs. This slide is just a summary of the data packages for the 2 growth hormone products that were initially developed. Both of these products involved direct chemical characterization of the follow-on products, the omnitrope and valtropin, to the innovators. They involve head-to-head pharmacodynamic testing. There was an extensive amount of animal toxicology. There were BE studies, bioequivalence, pharmacokinetic studies, although it should be noted that there's the follow-on products, the biosimilar products, were approved without the same level of bioequivalence as would be required for a generic drug. There were a number of additional pharmacodynamic measurements that were made, and for both of these products, these are other hormones like insulin, like growth hormone. The clinical data that were acquired for these development packages were extensive. Omnitrope, for instance, had 9-month direct head-to-head comparison with genotropin. There was switch to a different formulation when there were some problems. There were additional 9 to 12 months of safety data that were generated, up to 3 years in some cases of safety data. Altogether, there was approximately a couple of hundred patient years of exposure that were involved in the clinical investigations of these products. And just one final point at the bottom related to immunologic evaluation of these. It's always important to evaluate for the development of antibodies against the therapeutic protein, and in fact, when they're found, which is quite frequent, determine whether or not they're neutralizing or not. If they're not neutralizing, they're basically irrelevant. However, in the case of biosimilars that are made in biological systems like E. coli and yeast, it was also necessary for the manufacturers of the biosimilars to evaluate whether or not antibodies were being formed against proteins or carbohydrates of the manufacturing plants, the E. coli and yeast themselves. In Europe, there's been a number of biosimilar guidances and concept papers. What we will see in the U.S. is still uncertain. Nick's going to talk about, Nick Fleischer will be talking a bit about the change in the law and what the plans are, but we're clearly not as far along in this country as in Europe. So following the U.S. approvals of valtropin and omnitrope, we have a better picture of where the biosimilars program is leading. As I just mentioned, there are quite a number of fairly detailed guidances in Europe. The U.S. is still pending. I think these will be necessary in order for companies to feel secure in their development programs. It's quite clear that highly similar rather than identical active ingredients are sufficient standard to determine sameness, the sameness between the biosimilar and the innovator. The products that have been approved so far have been head-to-head comparisons, so there's not the issue of reliance upon proprietary data that would be found in an NDA or a BLA someplace, but direct comparative data. This makes some of the comparisons simpler, others perhaps not so simple. It's very clear that manufacturing processes do not need to be the same. In fact, the biosimilars approved today have not been made in exactly the same biological manufacturing systems as the innovators. Formulation differences are allowable as long as they don't impact on stability, immunogenicity, and other important factors. And in terms of the non-clinical evaluation, especially animal toxicology, which can be quite expensive. It seems clear at this point that some of this work will probably be required for products in the future. Others of the standard toxicology and other non-clinical evaluations will probably be referenceable. So finally, for my last slide in my section, I think what we've learned in the past few years with the approvals of the biosimilars is that the concept from a decade ago of a biogeneric is no longer there. There is no such thing as a biogeneric, and that whole idea has been replaced by the concept of biosimilar. Although the idea had been that these biosimilars might have abbreviated development programs, abbreviated meaning faster, cheaper, and therefore less cost to patients. The abbreviated programs have, in fact, themselves been very, very extensive. The databases really look like innovator NDA databases. Surprisingly, I think a lot of folks did not really predict this, but clinical evidence showing that a biosimilar product has a comparable therapeutic profile to the innovator product is actually quite difficult. These are biological assays, growth hormone, it's height change and things like that. It's actually quite difficult to show that your product is really the same biological therapeutic capabilities as the innovator, and in fact, it may be harder to show similarity than to show effectiveness in a standalone de novo BLA program. Nick will talk a bit about substitution and therapeutic equivalence. I would just say at this point that I think this is a very difficult area. FDA evaluations, I think, are going to be very product-specific. It's going to be necessary to have many, many meetings with FDA to determine what data need to be generated, what the comparisons should be, and what the requirements will be. And although we've checked and we do not actually have numerical evidence of what the biosimilar products have been sold for around the world, the initial suggestions from media sources is that the discounts that have been seen with the biosimilar growth hormones in particular have been in the range of 20 or 25% compared to the cost of the innovator proteins. This, of course, is in pretty sizable contradistinction to cost savings with good old-fashioned generic drugs that can amount to 90% or more savings with the generic versions. So I'm going to stop here. I'll turn the presentation now over to Nick Fleischer, and he's going to discuss a number of the more regulatory issues.
Well, thank you, Bob, and also I'd like to express my thanks to everybody that has joined our presentation today. What I will be talking about, and you may hear some repetition between some of the things that I will be presenting and what Bob has presented, and we felt that the repetition of some of these things are probably very important just to drive home some of these points that we'll be making today in our presentation. So I'd like to start with the regulatory history of where biologics originated from and their approval process, and I think we have to go back and just recognize that biologics were originally and still are approved under a different set of regulations and laws than the common small chemical molecules that we're aware of. Whereas the Food, Drug, and Cosmetic Act is what most pharmaceuticals are approved under, the biologics are approved under the Public Health Service Act. The main difference between the Public Health Service Act and the Food, Drug, and Cosmetic Act is that in 1984, with the passage of the Hatch-Waxman Act, it opened up the regulatory pathway for generics of small chemicals that were originally approved under the Food, Drug, and Cosmetic Act. However, there is no such act until recently for allowing a biologic that was approved under the Public Health Service Act to use an abbreviated pathway. However, we do have some sort of a precedent for addressing some of the scientific issues, and this goes back to a guidance that was published by the agency back in April of 1996, which happens to be a guidance that I was part of while still at the agency, which dealt with some of the issues that we're seeing today as being important to the biosimilar programs and what may be required for getting a biosimilar approved. As some of you may have seen this guidance, and it's still around, it basically called for analytical testing, it called for bioassays, it called for preclinical animal studies, it also called for clinical studies, and then there was a paragraph at the end of this guidance that said additional considerations, and as you can see it on the slide, the statement there was that the ability of the manufacturer to use validated and sensitive assays to demonstrate a product's identity and structure, biological activity, and clinical pharmacology provide a basis for determining whether comparability can be established without repeating clinical efficacy studies. Now, I must point out that this guidance, when it came out, was it may have been, somebody's back of their mind, a precursor of thinking about generic biologics or biosimilars. But at the time that it was really coming out it was the same time that on the drug side, the SUPAC documents were being developed, the scale-up and post-approval documents were coming into vogue, and somebody thought that we should also have something very similar in the biologics area when manufacturers of biologics were making changes in their products and how they would show comparability between an original product and a modified product. So, but the important thing to point out here is that guidance had a lot of the issues that we're seeing and possibly going to be seeing moving forward in how one would actually look at the scientific justification for approving a biosimilar product. As Bob mentioned, there are currently some products that have been approved that are, for lack of a better word, masquerading as biologics, even though they are approved under the Food, Drug, and Cosmetic Act as NDAs, and this being the human growth hormone and insulin products. Both of those products originally were grandfathered in under the Food, Drug, and Cosmetic Act. However, with technology and the use of recombinant technology mainly, these products would probably, if they came in today, would very likely have to go in through the Public Health Service Act because of the way they're manufactured. However, they were approved as NDAs, which also made them eligible then, because they were approved under the Food, Drug, and Cosmetic Act, to be filed as an ANDA, a 505J application, as a true generic application. However, as Bob pointed out earlier, at least the two examples that we have right now, which is the human growth hormone ones, both of those products went through a 505B2 NDA pathway to get their approval. As it's stated, there is no set criteria on an appropriate data set to support the approval of these products, and they were evaluated on a case-by-case basis. And as Bob pointed out in his slides, in his presentation, the data packages were fairly extensive for getting those products approved. Now, what has happened, two things have really changing the focus of the landscape, where generic biologics or biosimilars or interchangeable biologics may be coming into vogue, are two main things. The first one really is the passage of the Biologics Price Competition and Innovation Act of 2009, which amended the Public Health Service Act, and the further clarification of the law in the Patient Protection and Affordable Care Act of 2010, which is also known as the Healthcare Reform Law. Both of those acts have basically enabled a regulatory pathway, which up until the passage of those acts, there really wasn't a true-cut regulatory pathway for the FDA to rely on a regulation that they could use somewhat analogous to the Hatch-Waxman Act to use an abbreviated pathway for getting a biosimilar approved. And the other major issue that has occurred is that while in the past People always pointed out the differences between the Center for Biologics Evaluation and Research and the Center for Drug Evaluation and Research, that there may be different standards for approvability in both the requirements and clinical database and chemistry databases that would be required. The FDA recently moved all of the therapeutic biologics from CBER over to CDER, so now they're all approved and reviewed and approved by the same people that would be approving the small chemical molecules previously. So going on to the new rules that we have in place, as we said earlier, the biologics that were approved under the Section 505 of the Act, which means that any products that were previously approved under the 505, the Food and Cosmetic Act, can still be filed as NDAs. However, if there is a Public Health Service licensed biologic, a biosimilar application must be filed as under the Public Health Service Act. 10 years after the passage of the Act, which means around 2020, all the NDAs for biologics will have deemed to be approved under the Public Health Service Act. So it'll certainly eliminate this dichotomy of having some products that look like biologics being approved through NDAs or through the Food and Cosmetic Act pathway versus others being approved through the Public Health Service Act. Some of the nuances, important points of the new Act, there will be only one reference product for a biosimilar application. And one of the important things is also that the reviewing division for a biosimilar application will be the same reviewing division as the one that handles the reference product. The reason that this is important, as many of you may know, that currently in the small chemical generic world, new drugs are approved in the Office of New Drugs, whereas generic drugs have the Office of Generic Drugs that are approving those products. Whereas with the new rules, the biosimilars will be reviewed by the same people that approve the reference product. And just to round out the new rule, the REMS that have come into vogue under FDA will also apply to biosimilars as they become approved. The requirement for biosimilar application, according to the new law, the biosimilar must use the same mechanism of action if the mechanism of action is known for the reference product. The conditions of use and the labeling of a biosimilar would have to have been previously approved for the reference product. So this is very similar to the current regulations for ANDAs, which calls for identical labeling, at least for the most part, to an innovator product. And following along with that, the similarity between the ANDA rules that we currently have as to what is in the new Biologics Act, the route of administration, the dosage form, and the strength of the biosimilar product must match the reference product. In addition, the facility in which the product will be manufactured, processed, packaged, or held, they must meet standards that are designed to assure that the biosimilar continues to be safe, pure, and potent. Bob alluded to the issue of similarity, and I just want to, from a historical perspective, touch upon this sameness issue because it's going to be very relevant, obviously, to a biosimilar program. And this is not an issue that all of a sudden evolved with the new passage of the new Act, but we have had this issue going back quite a few years, where actually a company took the FDA to task and sued the FDA over the interpretation of sameness. As it turned out in that situation, after appeal, the D.C. Circuit Court upheld the FDA's interpretation of sameness, which is an important point because in many situations when it comes down to litigation in the courts between an FDA decision and somebody making allegations that the FDA scientific view may not be the most appropriate or correct, the courts have almost traditionally deferred to the expertise of the FDA in their scientific judgment. In that previous situation, the interpretation of the court of the statute that said sameness, it said it did not require same as to be defined as complete chemical identity. Even though, in the small chemical world, we do think of complete chemical identity, it's interesting to note that even in the Food, Drug, and Cosmetic Act, there is some latitude to say that it didn't require complete chemical identity. As I mentioned earlier, rarely have the FDA scientific criteria been questioned by the courts. And the key issue that will be coming up in the biosimilar area is that the criteria needed for determining how similar is similar enough when it will come to substitution of these complex products in clinical practice. So this will be a key issue that definitely will be a focal point for FDA to focus on. Because of the complex nature of these biologics products, it'll be very difficult for a one-size-fits-all systematic assessment of biosimilar to be utilized by the agency in approving these biosimilar products. Now, there has been some talk about, because of the complexity of the biologics and the biosimilars, that whereas, as I mentioned earlier, the courts have usually deferred to FDA scientific opinion, there certainly could be a concern in the courts that because of the complexity of these products, some judges may ask for an independent outside expert to help in explaining or resolving some of these complex issues when it comes to defining the sameness of a biosimilar product. I think we'll have to see that when it comes down the road, whether this actually will happen or not, but certainly we need to be aware of it, that the complexity of the biologics may not hold true for the same situations that we had in the past with deference to the FDA scientific opinion in many cases. And I'd also like to add at this point, I think for many of you that may recall all the litigation that occurred after the passage of the Hatch-Waxman Act, I can foresee that with the Biologics Act that was just passed, I think lawyers will definitely have a lot of work ahead of them in the litigation that may ensue when FDA makes certain decisions about the approvability of certain biosimilars. Which brings me to the topic of substitutability. I think this is probably the core issue that everybody in the biosimilar world is really interested in, is that how will substitutability occur? Now, substitution obviously is the core of the classic generic business model, because the way FDA enacted the Act and some of the analogous procedures that went along with it, such as the Orange Book with an AB rating code for products that are approved, and basically designating those products as completely therapeutically equivalent to the innovator product, set up a business model for generics that they don't have to market their product, and automatic substitution in many cases will occur as soon as a patient presents their prescription to a pharmacist, and will be given if a generic is available for that product, will be given a generic. Certainly it would depend on the therapeutic equivalence determination that has been made, but after the passage of the Waxman-Hatch Act in 1984, the FDA is prohibited from approving a non-AB rated generic, so everything that is approved post-1984 in small chemicals has to have that AB rating or substitutability rating to the innovator product. As mentioned earlier, the generic business model allows for a minimal sales force, and basically price, as mentioned by Bob earlier, price will be the driver, and there are multiple generics that come out, and it just drives the price down because it becomes a commodity issue. In the biosimilar world, what we may see is a substitution may evolve, but on a very limited basis. While the new law allows for interchangeability, it does not require a biosimilar product to be approved to actually seek an interchangeability designation by the FDA. And has been stated by Dr. Janet Woodcock, the currently head of the Center for Drugs, one of the ways that interchangeability will occur is that there has to be some sort of a switching study that will show that there will be no adverse events when interchangeability of a biosimilar product occurs. So the business model probably will not be identical to what we see right now in the generic world, but it will because it will not be a commodity. So just to summarize here, in a generic biosimilar world, when these products do get approved, and I think we had that example from the two the two growth hormones that have been approved, the omnitropin and valtropin, as they are not AB rated to their respective innovator product, they do need to be marketed. And the question will be, and I think Bob alluded to this, about the cost savings that we may be seeing. Will price be enough to drive utilization? And will there be resistance from physicians and patients in getting a biosimilar product instead of the innovator product? So moving on to interchangeability, which is probably the most important aspect of the whole biosimilar world, as I said earlier, is not a requirement to get a biosimilar approved. But to get interchangeability as a designation, the drug must be biosimilar to the reference product. It needs to have and expected to produce the same clinical result as the reference product in any given patient. And the way that this is going to be done is if the biosimilar is administered more than once to a patient, the risk in terms of safety or diminished efficacy of switching between the biosimilar and the reference product should not have a greater risk than the risk of using the reference product without switching. The big question is, how is somebody going to do this? What kind of multiple switch studies will be required? And these are totally unanswered questions at this point. We can't even try to fathom what kind of studies will be required to actually get a biosimilar product, even though it gets approved as a biosimilar, to then get that interchangeability designation. Just to quickly summarize the differences between small versus large molecules. A small molecule, as we know, they're therapeutically equivalent. It's the same molecule. It becomes substitutable under state pharmacy laws, and obviously the FDA's Orange Book listing of the AB rating goes a long way in assuring people that these products have met certain criteria to the FDA's satisfaction that these products are completely therapeutically equivalent to the innovator product. The price obviously drives these things, and as multiple generics get approved, the price will go down. Marketing, there really is no marketing for small chemicals per se, and obviously it's the cost that will sell the product. So generic companies have little need for formal sales and marketing staff. And the legal pathway, as I said earlier, was very clear with the passage of the Waxman-Hatch Act. It made it possible for a generic approval process, smooth generic approval process. In contrast to the biosimilars, we may not have therapeutic equivalence, and as Bob pointed out earlier, while the biosimilars will be so-called similar, they may not be exactly the same molecule. As I said earlier, the interchangeability will only come if the FDA finds the studies that will be required to satisfy the interchangeability concept to be satisfactory. And the question comes up is, will FDA have a similar type of a rating scale that they have in the Orange Book for drugs right now? Will there be a listing that will then identify which products have actually been designated as interchangeable? The price difference is obviously going to be smaller. It will require professional sales forces and marketing to drive utilization against the brand product. With the passage of the Biologics Act, the legal pathway obviously became clearer. The use of the 505(b)(2), at least for the growth hormone we've seen, has been utilized effectively to get those products approved. Certainly it may be a possibility for insulin products to get approved by that through that pathway, at least for the next 10 years. The biosimilar pathway with the amendment of the Public Health Service Act, with the new act, the Biologics Act, will enable biosimilars to be approved, but there's always a possibility of going to a full BLA if somebody doesn't want to undertake any other pathway in the foreseeable future. The marketing challenges, as I already mentioned, that for a classic generic, it's the substitutability that pushes sales. For a biosimilar, even though it may be the so-called generic, they will have to go out and detail them. The cost will be higher, and certainly this is not traditionally the sweet spot or the business model that generic companies have been used to. And the question will be, as I think we saw with generics coming into vogue, and certainly we've seen a great increase in the use of generics through the years since 1984. The question will be, how long will it take for an acceptance both by doctors and patients that these biosimilar products can be substituted for innovator products? It also brings up the point about what will innovators need to do to possibly distinguish themselves from the so-called biosimilar products. So it may require internal and external pressure for outcome studies to show if there are or are not differences between a biosimilar product or not. Now, what we've seen is evolving is new terminology. We've seen words like fingerprint-like technologies, and these will determine the product similarity. They could also limit the number of animal and clinical studies I mentioned earlier in the comparability protocol of 1996, where it said if you've got good data that shows comparability, then you possibly could limit the number of animal and clinical studies that would be required. Another term that we've seen thrown around is totality of the evidence, and I will get to these later of where these have come up. And certainly this is not a new concept. I think the FDA has traditionally, in approving drugs, has relied on integrating various kinds of evidence in making their regulatory decisions. So it isn't one piece of evidence, that it's a total package that they look at when they approve these drugs. And just to quote something from the FDA, that it seems possible to exceed a current state-of-the-art analytic characterization by evaluating more attributes and combination of attributes at greater sensitivities with multiple complementary methods. So again, just to reemphasize this, that it will be a total picture that the FDA will look at in making some of these decisions. Now, along with new technology, and new terminology, we've seen new technology. I think we've seen some of these things utilized, and to basically, these will help in the biosimilar characterization, the physical-chemical characterization, and I will not even go into describing some of these. I just wanted to present these as some of the newer technologies that are available out there that will be relied on both by companies that are trying to show similarity of their product to an innovative product, and will be probably required by the FDA to utilize some of these technologies to show comparability, at least in physical-chemical characterization, between a biosimilar product and an innovative product. And where some of this has come from is FDA's approval of a generic enoxaparin. Now, while some people may have said that enoxaparin is a pseudo-biologic, certainly it was approved as a drug through the Food and Drug Cosmetic Act. And what FDA has explained in their rationale for approving a generic enoxaparin is that they relied on 5 criteria or standards of identity, and they said those were taken together were sufficient information to conclude sameness. So I think there may be a lesson to be learned, even though we've heard other ways that some people have said that don't learn from enoxaparin FDA's approval, that it's not a pathway for biosimilars. But I must personally admit that what I've seen in that certainly, and a lot of other publications that have come out, certainly leaves credence to this totality of evidence that was used in the approval of generic enoxaparin. And the things they looked at, the equivalence of the physical-chemical properties, the equivalence of the heparin source material and the mode of depolymerization, the equivalence in the disaccharide building blocks, fragment mapping, and sequence of oligosaccharide species. And those 3 bullet points goes back to the previous slide about the new technology. I think some of that new technology really enabled both the applicants and the FDA to say that there is similarity in characterization between the innovative product and the generics. And the other things that were used, the 2 last bullet points on that slide, the equivalence in biological and biochemical assays, and the equivalence in vivo pharmacodynamic profile. Just quickly for contrast, because it certainly, at least in my mind, seems to have a little bit of a dichotomy, even though in FDA's explanation in their citizen petition response, they alluded to the fact that conjugated estrogens was not a similar thing to the way they approved enoxaparin. And before I go any further on this slide, there is a typo. The last bullet on the left side, it says 2 AB-rated generics. That should be AP-rated, not AB-rated. And that's important to note because the AB rating is given to a product that has gone through bioequivalence testing or bioequivalence demonstration to show their bioequivalent. In this situation with the AP rating, it was other methodologies that the FDA used to consider these to be substitutable parenteral products, which is what the AP rating stands for. So quickly to run through this slide as a comparison between the pathways that FDA used in approving enoxaparin versus conjugated estrogens. As we know, enoxaparin is just a heparin molecule that's chopped up into shorter chains, and as stated by the FDA itself, the product itself is not fully characterized, as can be seen by the molecular size distribution. There are ranges involved, so it's not one set molecule. There is a USP monograph for the product, and as I said earlier, there are currently 2 AP-rated generics approved for that. The conjugated estrogens goes back to 1942 as the original approval. Premarin, which is the innovative product, and there have been several follow-on products to Premarin since that time. It is a mixture of conjugated estrogens that are blended, and FDA took the position when generics of conjugated estrogens wanted to come out that the generics had not been shown to contain the same active ingredients and therefore to work the same. And as stated, the active ingredients in Premarin have not yet been confidently defined, and for that reason, FDA did not approve a generic version of the product. There is a USP monograph for conjugated estrogens, but at the current time, there is no AB-rated generic. And the next slide of something that happened very recently, and it's not to point the finger at Johnson & Johnson, even though they've probably taken their lumps and bumps over the past year or so with recalls, but the reason I point this out is here we have a biologic product. Eprex is not marketed in the United States, but it's an erythropoietin that is marketed outside the United States. And just recently, Johnson & Johnson had a recall of the product due to inconsistent potency of the product. So even in this 2011, we've got all that technology that I mentioned earlier that should be utilizable for characterization, especially when you come to measuring potency. Problems still can occur with products, and we've seen this recent recall of a product, of a biologic product. So moving on to the best practices in pursuing biosimilars, I think there has to be a defined regulatory strategy is very critical. I think it will require a very detailed data package. And one of the things that comes through very strongly in speeches by the FDA and publications by the FDA, that there will need to be a high level of collaboration with the FDA. What the FDA will require and will make the biosimilar pathway a lot easier for applicants is to provide the FDA with a completed, in-depth, comparative and analytic characterization and in vitro data. And based on what will be presented and the quality of that data, the FDA will be able to give the best advice on the scope, if any, required animal and human studies would be required. I think what we're going to see is a little bit different procedure than we've seen with the currently NDA world. There will be a need to communicate often and about everything. It may require more pre-IND meetings. It may require more IND meetings. One of the things that also that we've heard is that the FDA may want the user fees to be paid earlier. Currently, user fees are paid with an NDA application. It's very possible that because FDA is going to be putting all this up front, evaluation and providing recommendations and advice to sponsors, they probably will want that user fee paid up earlier to cover some of the cost of their additional work. And one of the things that I think people can certainly use is any precedents that are very applicable. So those should be used to the best advantage. Probably for these applications there will be a longer review time, and it may be one of the reasons why the FDA is thinking of or asking for an extension of their user fee goal dates from the current 10-month to a little longer time. Now, one of the interesting things is that the user fees for biosimilars will probably be identical to a full BLA. So there's certainly this concept of the user fee as being less we don't think it's going to be evident for biosimilar pathway. And something that you definitely have to be taking into consideration is to plan for scientific challenges that will come from the innovators. Obviously, the FDA hasn't defined the process yet. Certainly, the passage of the Biologics Price Competition and Innovation Act certainly helps because it now provides a regulatory pathway. The FDA has now been given greater definition, flexibility, but the problem is that there is no definition yet of what the appropriate data set is going to be. I think at the present time, and as we're hearing, it's going to be on a case-by-case basis. And there are a lot of issues. I think Bob alluded to these earlier, and these and are involved with the active ingredient itself, equivalence issues, substitutability issues, certainly marketing of a product, especially when it's not interchangeable, the pricing issues, and last but not least is certainly reimbursement issues. And the latest news that we have that just appeared a couple of days ago, courtesy of Reuters and other publications also, that the FDA is expected to issue guidelines on biotech medicine generics pretty soon. And Janet Woodcock has alluded to the issuance of the guidance within weeks and maybe even days. And interestingly enough, another FDA senior staffer, Dr. Roth, implied at a recent meeting that the concept of a biosimilar has been elucidated or at least alluded to in FDA's publication of an article in the New England Journal of Medicine. And just to conclude my presentation, I'm just going to quote out of that it says, although additional animal and clinical studies will generally be needed for protein biosimilars for the foreseeable future. And I cut that off at that point because I think the key word here is generally. So I think, as I said earlier, this is going to be a case-by-case basis, and we'll just have to see what the FDA's guidance will be and how FDA will start approving some of these biosimilar products now that they have a regulatory pathway for doing that. So that completes my presentation, and I'll turn it over to Mr. Swit.
Thank you, Dr. Fleischer, and also Dr. Roth, and to all of you that have stayed with us. We typically have a number of questions, and today is no exception. We are running towards the end of the hour, so we're just going to have a time for just a couple questions verbally, and then all of the questions that have been submitted will be answered at by written answers and questions within a week of this webinar. So. The first question, and I'll send this to Dr. Fleischer. Can I file a biosimilar application under the new law for a human growth hormone product, or must I submit it as an NDA? What's the status now?
Well, the answer to that question, under the new law, and again, this is probably going to be an area for lawyers to be more involved in, regulatory scientists possibly, a biosimilar application can only be filed for a biosimilar version of a biologic that has been approved under the BLA. So until an innovator secures a licensed BLA for a human growth hormone product, that product would have to go through an NDA process at the current time. However, after March 2010, all of the biologics that were approved as NDAs, such as growth hormone, such as recombinant insulin, will automatically consider it as BLAs, and at that time, the biosimilar application pathway would kick in.
Dr. Fleischer, did you mean 2010 or 2020 on that?
I'm sorry, 2020.
Thank you. And one last question before we will adjourn. Also for Dr. Fleischer, the exclusivity provisions of the new law give innovators 12 years from the date of approval of the innovator's biologic. In the generic world, we saw lots of evergreening attempts. Do you think innovators in the small molecule world, do you think innovators will try to evergreen their products by making small changes to secure new BLAs to extend get new exclusivity periods?
This is a very interesting question because it's very possible that things like this could occur. I mean, we saw this with small chemical molecules until FDA and their laws were changed, the regulations were changed. But I think what we possibly could see in this area is that innovator companies could potentially do minor changes to their product. I think we have an example that inadvertently, I think, when Genzyme went to scale up their Myozyme product, FDA concluded that the product was not the same as the previous product that was manufactured, and in fact considered the product as a new product. So whether something like this is a pathway that innovator biologic manufacturers may consider as a way of evergreening, it is possible. But I think certainly it can happen. I think we'll just have to wait and see when some of these things occur, how they will pan out.
Well, thank you, Dr. Fleischer and Dr. Roth, again, for sharing your insights with our audience. And on behalf of the Weinberg Group, I'd like to thank all of you for taking the time out of your day, no matter where you are, whether this is morning or even evening where you might be. We do have a survey that we would appreciate you completing as to your impressions of today's webinar and any suggestions you might have for future topics. As you may know, this is a recurring series of complimentary webinars that we sponsor, usually monthly, of topics of interest to the life sciences world from a regulatory perspective. So I thank you again for attending and wish you all a very good day or evening wherever you may be. Thank you.